Hi everyone, welcome to the first talk of the last day of the conference. I hope you've had a good time so far. I'm here today to give you a talk about a part of my PhD project in Scotland, the adaptive landscape of the endangered freshwater pearl mussel, also known as the eastern pearl shell in North America. So margaritifera is a widespread mussel found around the Holarctic region. It has an obligate parasitic stage, but before I get into that, I wanted to give a fun little side note. Before the trade routes opened up across Europe into Asia, it was the primary source of freshwater pearls, or pearls, for the European people. So on the oldest crown jewels in the UK, you can see pearls, which is really fun. And also in about 74 AD, Pliny the Elder wrote about Julius Caesar's love of freshwater pearls. Which is probably one of the oldest written documents of animals ecology in the UK, which is pretty cool. So they have this obligate parasitic stage where the mussels release these Glaucidia larvae into the water column and inhaled by these salmonid fish when they insist on the gills. So that's them on the gills, and they stay there for about 9 months before they then drop off into the sediment of the river and develop for about 5 years before they come out the sediment and reach sexual maturity about 12 years old. So they have this really extended life cycle. And they are classed as endangered in Scotland. In the UK, they're critically endangered. And the major threats are pearl fishing. So 1 in every 100 mussels produces a pearl. So if someone does find the mussels and they're interested in pearls, they can come in and wipe out a whole bed of mussels in one go. So that's a big problem, especially in Scotland. They're also impacted by lots of other things like pollution, river engineering. Obviously, if the host fish disappear, then they can't complete their life cycle, and climate change, which is obviously a massive issue at the moment. So, as they have such a long life cycle, they are very much reliant on things like plasticity or their standing genomic variation to sort of adapt. So to detect things like that, you really do need this fine-scale genomic research. And generally, in my literature review, a lot of previous research in margaritifera has been more at the microsatellite scale, and I found 2 papers that produce fine-scale research. So this one in central Massachusetts, looking at 6 sites across 2 rivers, which show low structure and diversity, and then another paper in the Iberian Peninsula that was across 26 sites across 16 rivers, and they found quite high population structuring, but low diversity. So I wondered what was the population structure and local adaptation in Scotland, which holds roughly half the world's reproducing populations of these really endangered mussels. So my research questions were, how are the populations structured in Scotland? How connected are these populations? And then is there any evidence for local adaptation which could potentially help with resilience to climate change? So I was very lucky to start my PhD with data already collected, as these mussels are so endangered. It's a challenge to even get in a river with a muscle in Scotland, you need a license. And I think there are about like 6 people in Scotland who have a license, and you need 2 of those people to sign off to get a license. So I was very lucky that my supervisor, Victoria Pritchard, had already had those samples collected. And these were collected using these non-invasive swabs, so none of the muscles were killed, thankfully, where you sort of pry the muscle open at the site, and you insert like a kind of like a COVID swab situation, and then you can extract DNA from that. So from those, and through RAD sequencing and like the bioinformatic processing, I ended up with 156 samples to work with across 18 sites and 5,486 SNPs. So for population structure analysis, I pruned for linkage disequilibrium and was using this reduced SNP data set of 3,486 SNPs. And I've presented these on PCA, which I'm sure we're familiar with at the end of the conference. But just to reiterate, each of these dots are an individual, and I've circled each of the river populations on that plot. And it's just to represent genetic differences over a 2D space. So I just first wanted to pull out this group on the right-hand side, and they are clustered on the Ardnamurchan Peninsula. Oh, I also wanted to say, as they're so endangered, you'll see a lot of plots. I can't plot exactly their location, so I have jittered these points 10 kilometers away from where they are, and it's also super zoomed out, so hopefully you can't figure out which rivers they're in. And earlier, that's why we had heat maps of vice counties. Yeah. So this is the Ardnamurchan population, and they are geographically clustered, but on the lower left-hand side, you can see we've got this really strong cluster here, but they are scattered across Scotland, which I find quite interesting. And we also have this cluster here, which is structuring kind of geographically, but also we have these populations out in population out in the Outer Hebrides, and then on the east side of Scotland, where the rivers discharge into the east, into the North Sea. So they might be geographically close as the bird flies, but as the fish swims or the host swims, they're quite far apart. So that's quite interesting. And I sort of, as a brief way of describing that, I have plotted FST against distance for you as well. So there is no correlation. So host use has been reported in the literature as explaining some of this variation. So it does here too, which is great, but it doesn't explain all of it. And I'm not going to focus this talk on that. I wanted to figure out what else could explain some more of the variation here. So there is a range of environmental gradients across Scotland. So we have, obviously, climate, the pressing concern, where temperature and precipitation varies across that range. There's also habitat values like altitude, pH. And as there's a long history of pearl fishing in Scotland, I thought it would be cool to look at remoteness. So remoteness from roads, and this is also a factor of ruggedness of the terrain as well. So that's quite fun. So to look at all of these different factors with the multivariate SNP data set, I used a redundancy analysis. And this is the result. The model was significant in, I think, all aspects, which was great. And I identified 249 putatively adaptive SNPs, so using the outlier approach. And these were mostly associated with altitude, elevation range, host, and then like soil parameters, wilderness, and temperature at the end. These outliers were distributed across the genome. So we also don't have we have this, it's not a model system, right? So we have the second reference genome, which was released last year, and the annotations isn't quite there at the moment. So this is across 156 contigs, and all of the SNPs in the data set are across 800 contigs, so a large proportion. Yeah, and as the annotation isn't there, functional inferences are not, I haven't managed to do that yet, but we are thinking about it. So, yeah, so next steps for me, in the next few months, I'm hoping to produce some kind of genomic offset analysis to identify maybe where there is a gap in fitness in space between these adaptive alleles. And I'm going to be looking at predicting drivers of decline, because I have this other really cool data set which shows where pearl fishing has occurred in recent years, and that kind of thing, reproducing populations versus not reproducing populations. So main takeaways from this talk today: populations are structured across Scotland and don't generally follow catchment geography. Host use does explain some genomic variation, but not all. Environment explains some genomic variation too. And most of all, conservation must consider population structure and local adaptation going forward. So with that, thank you very much to my supervisors, Cara Layton. I know she's recruiting for her lab in Toronto. And also Victoria Pritchard, a shout out for her, because she also collected the majority of these, all of these DNA samples. So yes, thank you very much. And do you have any questions? Hi, yes.
Is there any transport history? I would think that they moved around and that the fish would bring the recruits back to the river.
Yeah, so I don't have that data entirely, but yeah, it's something I'm very much considering, looking when I delve further into this DNA. Hi. Yes?
Is there any seasonality or temporal differences in when these mussels are infecting the hosts, and can you explore that with some of the environmental features?
I think there is definitely seasonality. I'm not entirely sure. Looking at the literature, I'm still sort of figuring out whether, I think there's a bit of contention whether we know exactly when the Glycidia are released. I think some people were pretty sure and some people aren't. But yeah, that's a really cool point. Yeah. Hi.
Very cool talk. What are some of the ongoing conservation efforts for the freshwater mussel? Is it really just no pearl fishing and protecting those areas, or is there any fishery supplementation if they're able to be brought into a laboratory setting or something like that?
I'm not sure about laboratory settings, but I know there are experiments where people are taking the host fish and collecting the Glycidia and try and help induce them to infect their host, and plenty of other things which I can talk about during the break. Yeah. All right. Thank you.
Hi everyone. My name. Oh, I went too far. My name's Greg Owens. I'm an assistant professor at the University of Victoria in Canada, and today I'm going to tell you about a new project in my lab looking at population genomics in kelp species. And before I talk about the results, I want to give a acknowledgement to all the partners we're working with. So a lot of people were involved in collecting all of the samples, including several First Nations communities who collected on their territories, as well as funding sources from MITEX and Ooh, this is not right. MyText and Genome BC. I also want to acknowledge Dr. Jordan Bamels, who is a postdoc in my lab and did all of the work in this project. I'm just a supervisor. It's all Jordan's work, and he did an amazing job. Kelp forests are really important marine habitats, so they occur on the coast. Kelp forests can grow up to, in areas up to 50 meters deep, so these enormous kelp species. The main species here are these canopy-forming kelp. In this photo, this is giant kelp, and it is enormous. It provides structure in the marine environment and is an important habitat for fish. It's also potentially important for carbon sequestration. So these plants or these algae grow quite fast and potentially are sequestering carbon. On the coast of BC, we have two species of canopy-forming kelp. We have giant kelp and bull kelp. And some recent work has looked at the sort of persistence of kelp forests over time. So this study by Sam Starko et al. Looked at kelp forests in different parts of BC and looked at whether the kelp forests had declined over time or are increasing over time. And there's a lot of sort of populations that haven't really changed, but especially in this sort of red area you're seeing in the Salish Sea, kelp forests are declining a lot. That's largely or partially because of temperatures. The Salish Sea is one of the hottest seas north of California, and also because of urchin predation. So our question coming in here is we have these kelp forests that are declining. We have ongoing restoration efforts. We took the sort of genetic approach of thinking about what is the genetic diversity of kelp populations there. So, and can we use what we know about genetic diversity to help inform conservation? To do this, we used whole genome sequencing. So we looked at both bull kelp and giant kelp, and we sequenced between 70 and 35 populations between the two species, basically just sequenced whole genome data. So jumping right into the results here, these are the populations of bull kelp and giant kelp. And I'm plotting here nucleotide diversity. So this is just sort of a level of genetic diversity. And for giant kelp, the populations are relatively consistent. They're all sort of at a similar level of diversity. But for bull kelp, we have a wide range of diversity. So we have about 40-fold difference in diversity from our most diverse to our least diverse. Highest diversity in the north in Haida Gwaii, and then lowest diversity down in Puget Sound in the southern populations there. One of the things we sort of found when we were analyzing this data is we could look at sort of heterozygosity across the genome. So this is a single sample and just looking at observed heterozygosity in windows. And we can see chromosome 18 to 24. And the heterozygosity goes up and down, but it's relatively high or consistent. But we also have samples that look like this, where we have high heterozygosity and then long stretches of virtually no heterozygosity. And this is really indicative of a selfed sample. So we are seeing here some samples that are first generation selfed. We also see, just for comparison, this is the Puget Sound sample, which we don't think is selfed, but just is incredibly, incredibly inbred. And we're sort of following up on why the Puget Sound population is still alive, because this is basically no diversity. There's almost no diversity left in this population. This population is actually quite small and is barely hanging on. So ways of looking for selfing is we look for runs of homozygosity, so stretches across the genome where there is virtually no heterozygosity. And we measured this using BCF tools as well as Rohan and got very consistent answers. So this graph is not a structure plot. The colors represent genetic groupings, and then the black bars represent the percent of the amount of the genome that is in a run of homozygosity. So for bull kelp, we can see most of the individuals have very low runs of homozygosity, but then there's every once in a while there's somebody up there at about 50%. For giant kelp, we have generally higher runs of homozygosity, indicating sort of more ongoing inbreeding, as well as some individuals that are, again, look like first-generation selfed. Overall, if we look at all of our samples, we see that about 10% of our individuals are selfed. So we then were thinking about this. Okay, we have these runs of homozygosity. We are interested in how good the populations are. Are they going to be able to be maintained over long periods of time? So we used a method out of Kim Gilbert's lab to estimate effective population size using runs of homozygosity. And this machine learning method simultaneously estimates a self-fertilization rate as well as a population size. So we ran this for both giant kelp and bull kelp. For bull kelp, our populations range from about 8,000 to about 30, so we have this wide range. Some of them are quite small. And for giant kelp, all of our populations are around 100 to 200. So the populations, even though this is a very wide-ranging species that has some of the giant kelp spans almost the globe, or not all the globe, but there's populations all over. Each individual population itself is actually quite small. These measures of effective population size are using the runs of homozygosity, so they're not directly using nucleotide diversity. And we see that there is a correlation between nucleotide diversity and the measures of population size, giving us some confidence that these are accurately measuring some sort of aspect of the effective population size. So we have these wide range of population sizes in bull kelp, and to a lesser extent in giant kelp. And we are interested in a question of, if they have these small populations and they are selfing fairly frequently, are they actually purging deleterious alleles? So we know that in almost all populations there are recessive deleterious alleles, and for an outbred population they're generally not a problem because they are found in the heterozygous state. But if we have an inbred individual, those those recessive deleterious alleles will often become homozygous and cause problems. And in some systems we've seen, or some systems other people have seen, that small populations can purge deleterious alleles. So they're very small, these recessive deleterious alleles are then expressed and then are removed. So to answer this, we used GERP analysis, or genomic evolution rate profiling. We aligned about 30 different brown algal genomes to the kelp genome, and we looked at all of the sites that align, and we sort of characterized them as evolutionarily labile or evolutionarily conserved. So a labile site often changes over evolutionary time, so this one is probably not super important to stay the same, but an evolutionary conserved site is one that hasn't changed at all over hundreds of millions of years of evolution, and if we see a mutation in a kelp individual, that likely means that it's probably not very good, it's probably a deleterious allele. And we also sort of polarize them as ancestral or derived. So our hypothesis here is that if only drift is happening, there's no purging, then the total frequency of derived alleles in populations is not going to change over population size. So small populations will have the same frequency of derived alleles. They'll have more homozygous derived alleles because they're small and inbred, but they will have the same sort of total number. If there is purging, then we expect these small populations are going to have fewer derived deleterious alleles. And we won't see the same pattern in the evolutionary labile sites, which are putatively sort of neutral. In our results, we see no evidence of purging. So for both bull kelp and giant kelp, there's no relationship between the effective population size and the total frequency of derived deleterious alleles, derived alleles, regardless of whether they're potentially deleterious or potentially neutral. So the next sort of question is, okay, maybe they are all purged. Maybe nobody has any deleterious alleles. We think that's unlikely. There's been some experimental work in the past in giant kelp that showed that selfed individuals have lower survival and lower fecundity. So this plot on the left is showing the fecundity of plants that are at different levels of self-fertilization. That being said, all of the plants we collected were adults, so the selfed individuals made it to adulthood, but we don't know if they are less fit. And this is some future work we're planning on to actually measure the difference in fitness between selfed and non-selfed. Lastly, I want to talk about a little bit of pattern we saw. In this case, we are looking at all of the populations color-coded by sort of genetic groupings, and we are showing the proportion of homozygous deleterious alleles. The bar charts, box plots are the outbred individual, and the shapes on the top there are selfed individuals in that same population. So across different populations will have different proportions of homozygous derived deleterious alleles. And we always see that the selfed individuals have more homozygous derived because they are selfed, they have more homozygous alleles overall. But there's a sort of an interesting pattern here that the difference between the outbred individual and the selfed individual varies in the different populations. So on the left here, there's a population that's where the outbred individuals have very low homozygous derived alleles, And then the self individual is much, much higher. So they have a much more sort of deleterious load, or realized deleterious load. And then in some populations, basically all of the individuals have a lot of homozygous alleles, and then the self individuals just have slightly more. We looked at this relationship. So the y-axis here is the relative difference in the individuals that are outbred versus individuals that are selfed. So a number higher than 1 means that the selfed individuals are much sort of worse, have more homozygous derived deleterious alleles. And there's a linear relationship here, so that the populations are very small, have very little difference between the selfed individuals and the non-selfed individuals. And we think this is really consistent with a sort of just simple genetic drift, in that small populations, many alleles are fixed, and they tend to fix even in the non-selfed individuals. So everybody seems fairly, looks inbred, looks very homozygous, and if you self, there's not much of more of a problem. But for our larger populations that are more outbred, selfing has a dramatic difference in homozygosity and dramatically sort of increases your deleterious load. For giant kelp, we don't see the same pattern, although we have a much smaller sort of range of population sizes, and it's possible we just haven't, they're not in the, perhaps with a larger population size range they would see it. But regardless, what we are seeing here is a species that has frequent selfing, but it hasn't seemingly purged deleterious alleles, even though it has selfing and small population sizes. And that this, in small populations, there seems to be less of a relative penalty of selfing. And this sort of suggests that maybe in these small populations, selfing is more of a better sort of reproductive strategy. Although the caveat here is that really we're talking about relative fitness here between outbred versus inbred. It's distinctly possible, and I would say probably probable, that in these small populations, everybody is doing pretty badly, and that if you are selfing, you're just not much worse than the background level. And I want to thank you for listening, and I'm happy to take any questions with my time. All right, speak now or hold your peace. All right, thanks everyone.
Hello everyone, I'm Chris Boccia, a PhD student in the Friesen Lab at Queen's University, and I'm going to be talking to you about the historical demography of northern seabirds and their prey, and assessing responses to past climate changes. So, as we all know, polar habitats are undergoing pretty profound changes. So this is one of my favorite places in Alaska, Northwestern Fjord. And so the first time that I went there in 2010, there were 2 tidewater glaciers, so glaciers that extend all the way down from the ice fields to the ocean. And then when I visited again in 2023, only a decade or so later, there was only one. So one of them had already receded so much that it was no longer classified as a tidewater glacier. And so yeah, it's pretty wild to see that kind of change just in my lifetime. And it's a large deal for a lot of seabirds in the area, especially Kitlitz's murrelet, which is sort of reliant on the interface waters between tidewater glaciers and the ocean areas there. And so unfortunately, we're probably going to see this happen a lot more. So these are just some future possible climate trajectories, depending on how well we do with reducing fossil fuels. But even if we do a really good job, the poles, unfortunately, are going to feel it a lot more than the rest of the world. So there's going to potentially be more situations like that, where there are large habitat changes and species have to figure things out. On the bright side, though, if we zoom out a bit, large changes in climate have happened in the past. So this isn't some of these species' first rodeos. So due to Milankovitch cycles, we've had really large changes in sea level and climate before. So just the present is in the center there, and the plot on the left has time on the log scale. And so, there is obviously some capacity in these species to deal with warmer temperatures, since every species that's alive today has had to go through this at some point in the past. So for conservation purposes, it would be really great if we had some way to kind of quantify this adaptive potential so that we could figure out which species to focus our efforts on. And so one of the more reliable methods to do this is common garden or reciprocal transplant experiments. So you take an organism, usually a plant, and you move it from a current environment to an environment that kind of matches what you expect the future environment to be. So unfortunately, this is great for plants and some more sessile organisms, but for my seabirds, it's not such a good option. You can't really tell a kittiwake just to stay in one patch. So that's out. And then you can also do long-term phenotypic studies with pedigree data. So if you have traits that you know are linked to climate survival, you can take a look at those and then figure out what kind of potential exists in the population. And there are some places where you can do this with seabirds. The Middleton Island research site is a really good example, where they do have a long enough term data set to be able to start looking at these questions. But unfortunately for the vast majority of seabirds, we haven't started collecting data far enough back to do that, or we just haven't had the money or ability to access their nesting areas. So that's not really an option either. So we're unfortunately left with indirect methods, so getting proxies for adaptive potential, and one of these is historical effective population size. So the idea here is if we could just go back into the past and look at how well species did during some of those previous warm spells, we'd have some idea, forecasting forward, how well they'll do under another warm spell. So of course, if we had a time machine, we could just go back and actually try and census the populations. Unfortunately, my time machine proposal was not funded, so I had to try something else. And so what I ended up using was the pairwise sequentially Markovian coalescent method. So for this, you use the diploid genome of an individual, you identify where all the heterozygotes are, and then you use a hidden Markov model that kind of tracks its way along the genome and identifies ancestral changes of recombination state, and those are the hidden states, and then the heterozygotes are the emissions. So it goes through, figures all that out, and then uses expectation maximization to establish the optimal stepwise function that best explains that Markov path. And this is just an example for the Pacific subspecies of black-legged kittiwake here. So I'm not the first person to have this idea. There was a paper in 2015 that looked at the species that were available then for birds. And so they found 3 broad patterns. They found that there were species that declined during the last glacial period, and species that sort of had a long-term decline, and then other species that sort of had cyclical patterns. And as well, there was a really complex and wide-ranging study in 2023 by Gillman et al. And so they used a cluster analysis to group similar demographies together. So I'm using some of the same methods, so I'll just quickly explain a bit of this plot, which is very complicated looking. So on the left side there, that's actually not a phylogeny. That's the hierarchical clustering, just to avoid confusion. And then for each of those bars, those are all individual species. The red is higher population points in species history, and blue are low population points. And so, so the method essentially just goes through and clusters all of those. And the way that they managed that was by having sort of the same sampling points all throughout to be able to compare the curves. So Gillman et al., they also identified some climate-sensitive species by assessing behavior in recent times of the curves. And then they used a phylogenetic path analysis to identify some traits that might increase vulnerability. So for example, species that were warming negative or vulnerable to future warming they found signal in incubation duration, size, and egg size as well, in terms of whether or not they would be sensitive or not. So sort of what I'm adding to this is a prey species aspect. So of course, how well seabirds do is partially going to depend on their ability to get food, and so no one's really looked at prey in relation to seabird curves before. And I'm still going to be reconstructing those curves, assessing similarities between the demographic histories, and then also looking at correlations between the population sizes and historical temperature and sea level. So I ended up with about 13 different species, if you count the Atlantic and Pacific black-legged kittiwakes as different. So kind of a nice mix of different foraging modes, some sort of like large predatory seabirds like the herring gull, some surface feeders like the 2 kittiwakes, 1 sea duck, and then a lot of diving feeders. And as for the prey, I was also sort of limited by what was available, but I tried to get as many as I could, sort of a range of species that are from both ocean basins and that are taken to some degree by some of these birds. Some of these you'll obviously look at and see are quite large fish, It's not the adult stage necessarily that's being taken, it's some of the younger, smaller stages. So I was able to download whole genome sequences from GenBank for this, and then I followed a general pipeline to do cleaning and trimming and filtering and variant calling. And then I ran PSMC using some of the experimentally derived best parameters by those previous authors. And then in terms of analyzing the curves at a post hoc after that fact, I used R to do the same sort of hierarchical clustering that was done by Germain et al. And then I used a linear model with a lag component to assess the effects of sea level and temperature on population sizes. So jumping into the clustering results. This is the same sort of plot that I showed earlier, just with a different orientation, where the present is close to the sort of phylogeny-esque clustering method at the top. And so yeah, again, once again, these curves are, the blue areas are low population size, and the red and orange areas are higher population size. So with this method, there seem to be sort of 4 general clusters. So I'll just run through each of those. So the first cluster here, and these are the PSMC plots that are a component of that. And so this one here, these are species that I would probably be most worried about going forward, just because for a lot of them, their lowest NE was during previous warm periods. So right before the last glacial period there, they all have a sort of a dip there, with the exception of a couple. And then they increase after that. So potentially some concerns there. And a lot of these were actually prey species. So we got, and as well as the red-legged kittiwake. So red-legged kittiwakes are unfortunately already somewhat of a species of conservation concern because they're only found on a few islands in the Bering Sea. And interestingly, it's a bit tricky to pull out without a laser pointer, but the red-legged kittiwakes actually followed pretty well the population trends of the polar cod and the Arctic cod, which is interesting. I didn't see a lot of correlation with prey overall, but that one seemed to be, the curves actually matched up pretty well. All right, on to the second cluster. So not a whole bunch of species in this one, just common eider and lesser sand eel and a possum shrimp. And so with this one here, it's a little similar to the other pattern, so it was associated with that, in that we do have that increase towards the present, but these ones here seem to have sort of a long-term decline before that increase. And so, yeah, I'm not sure entirely what to make of that in a conservation sense. Obviously, a long-term decline is not a good sign, but it doesn't seem to be linked to temperature, so there's perhaps not a reason to think that they'll do any worse than anything else. So the third cluster was by far the largest, and so this one actually kind of matched up well with one of the clusters found in that 2015 paper. So these were all species that experienced a decline starting pretty much right when the last glacial period began. So presumably maybe ice over nesting sites or something like that caused this. And so yeah, we had species from both basins and sort of multiple, pretty much all feeding modalities, and as well as a few prey species that showed that general pattern. So it's hard to know, maybe these species will actually be okay with climate change just because they were seemingly doing okay during warmer periods before and just crashed with that glaciation. All right, and the last cluster here. So I think this one also does match up with one of the clusters from that 2015 paper. These are species that seem to have really cyclical demographies that go up and down. So hard to figure out any conservation trends from that, just since they seem to be pretty variable. And yeah, again, sort of a distribution of species, not really from any one basin or feeding modality. And so yeah, I also looked at sort of the overall effect of temperature on population size across all of these species. And yeah, I had to do a little bit of autocorrelation correction because of course, it's not too possible for most species to instantly change their population too far up or down. And so when you took all that into account, there was a very small actual increase in overall population size with temperature in seabirds and a very small decrease in prey species, which I wasn't necessarily expecting, and they didn't match up was also interesting. So again, these results are, I finished these like 2 days ago, so I wouldn't put a huge amount of confidence in them, but that's at least the preliminary result for that. And sea level had the same trend. Of course, sea level is highly correlated with temperature, so that's not really very surprising. So just some overall take-homes. So seabird responses to climate change seemingly vary quite widely. So there are some species that maybe we need to be concerned about, and then some other species that seemingly might be doing okay. Looks like there's some evidence that prey populations may decline with climate warming, which is not a good sign given sort of the attacks we're already making on prey species overall. And I think seabird survival as well will likely depend more on the other impacts overall that we throw at them. So, like bycatch, hunting, stock depletion, and nesting site destruction than just climate alone. So seemingly they have some ability to make it through warmer time periods, and it's sort of if we throw too many other challenges at them that they might not be able to figure it out. Yeah, so I'd just like to thank my funding sources and everyone in my lab, and as well the Digital Research Alliance of Canada that provided the compute power to run some of these pretty intense analyses. All right, thanks, and I'll take any questions. Sure.
Thanks for the talk, it was really interesting. I was wondering, with using effective population size as the proxy, are there any confounding variables that you might expect, like complicate that relationship between temperature and population size?
Yeah, potentially. I mean, part of the issue with effective population size and these trends in general is just that structure can kind of confound them at certain points. So that's definitely, especially when you do a really wide-ranging study like this, it's definitely possible. Yeah, but there definitely could be things that confound it, but it's just sort of what we have for this kind of thing. Yep.
For the increase in seabird effective population size and the decrease there in the prey item, are those, I guess, two questions. First, like, are those coupled such that like that prey item is like respective to like that bird that you showed?
Oh no, that's across everything. Yeah, I wish I had that kind of exact matchup data, but
Okay.
yeah.
Well, then, so follow-up question then is, well, like the way I guess you framed it. So like if the food source was declining, that wouldn't lead to like seabird population increase. So instead, like, what do you think relates with temperature and like seabird population increase, like contributing to prey food decline?
Well, I mean, I'm not necessarily sure that the seabird increase is causing the prey decline. I think they might. I think seabirds, it could be coupled to that, but I think seabirds are pretty good at switching prey sources to whatever is available, and so that might be partially why we don't see a lot of really strongly tied seabird species to prey. But in terms of the seabird increase, I think part of it might be driven by just nesting site availability. I think they have seen that. That's been noted in albatrosses specifically. Someone did the modeling to look at what areas would have been ice-free. And I think that might be part of it, just because, yeah, we had, especially with the species that I had, so many of them declined, like with the last glacial maximum. So that's probably why there's that overall trend, just all those species driving it.
Curious, so for your cluster one, it looked like the effective population size prior to the last interglacial was lower than the modern effective population size. Is that hopeful? I mean, could there be more adaptive potential now to deal with warming climate?
Possibly. I mean, the unfortunate thing with those curves is just that the uncertainty increases closer to present. So I'd like that to be true, but yeah, I'm not totally certain. All right. Thanks.
So, hello everyone, my name is Ryan. I'm a fourth year PhD student. I'm actually finishing in a few months, so arguably I shouldn't be here and I should be writing my thesis, but that is a question for a different time. Today I'll be talking about the interplay between transposable elements and domestication. So some people might think it's a bit of a niche kind of convergence of two different themes, and that is correct. But actually, the context of one provides quite a good kind of environment for the study of the other. Both are quite growing fields, and we're learning more about like the fundamentals about transposable element evolution, and also kind of the processes behind domestication, especially the genomic bases as well. So just a quick primer on transposable elements. I'll keep it brief because this is the genomics session, so I'm sure a lot of you already know about transposable elements. So the kind of key things are that they are mobile genetic elements. They can move around in the genome and can also propagate at the same time. They're also selfish, so that they can replicate at the expense of the host, so regardless of what is, selectively beneficial for the host. And we think about a generalized kind of structure for them. They have a variable number of protein coding regions, which are used in transposition. They can also have none, and then in that case, they kind of rely on other TEs in the genome. And they also often have repetitive elements in their flanks, which are used as identification sites for their propagation. So transposable elements are very diverse. The main dichotomy is between class 1 and class 2. So class 1, all you need to know really is that they use an RNA intermediate and they use a copy and paste style transposition, and class 2 use more of a cut and paste style. And you don't need to know much about the structure, but just that there are 5 major groupings that I'll be discussing in this talk. So those are LTR lines, signs, DNA elements, and helitrons. Transposable elements are also ubiquitous, so they're pretty much found in all eukaryotic genomes, and they're even found in bacteria and the genomes of some viruses. In some of the talks that I've been here, actually, I've seen that people who haven't been studying TEs directly have actually found TEs affecting their study system, which kind of just emphasizes how prevalent they are and how much they can affect pre-existing pathways and genomes. And so you'd expect that TEs can have adaptive effect or disturb genes and existing pathways that result to changes in the phenotype. And that is exactly what you get. So kind of growing evidence shows that TEs have contributed to a lot of different phenotypes. For example, coloration, there's a good review out by James Galbraith, who used to be in my lab, reproduction, behavior, growth, immunity, just to name a few. And these categories are also they're very relevant for domestic phenotypes. So you want increased growth because you want increased meat production. You want more nice animals that you can have as pets. And so that already provides sort of prior for TEs to be affecting domesticate phenotypes. And you do get some examples of that. So for example, in horses bred for racing, there's a sign that's known to increase muscle production that has increased racing performance. And then in dogs, there's lines or signs that have led to a change that people think might lead to more amicable social behaviors. So we see that TEs have affected the phenotypes of domestic animals, but we don't know to what extent this happens. And so part of my thesis is trying to uncover how TEs might contribute to these new domestic phenotypes and the process behind it. But why should we care about domestic animals? So in modern populations, they really underpin a lot of aspects of our society. So for example, livestock and poultry species, such as pigs, cows, sheep, and chicken, they provide very integral sources of food, especially for a growing global population. And then we also have pets that provide companionship, beasts of burden that provide kind of manual services, and other animals too, such as silkworm, which provide other useful commodities. Now, domestication is also very interesting evolutionarily. So the process has allowed human societies to transition from more hunter-gatherer lifestyles to more sedentary populations that can then accommodate large populations and more complex societies. And obviously, of course, that has domestication has allowed for rapid and quite drastic changes to animal phenotypes. So for example, if you take a wolf and a chihuahua, you probably wouldn't really put those two together. And on the other hand of that, domestication is also a very interesting context to study TEs and the evolution of TEs. So archetypically, domestication involves various different environmental and demographic factors. For example, many domestic species have experienced severe bottlenecks, leading to much smaller populations. And then that allows you to ask, what effect does population and therefore kind of the selection drift balance impact on TE content composition? And also, obviously, there's a very different selective regime. So when domestic animals are taken from the wild, the reduced purifying selection is then reduced, and they have kind of human selection pressures, which can then vary in strength. So this kind of intersection allows us to ask really fundamental properties about both of these kind of research themes. So my kind of research questions are, how does domestication affect TEs? And then how do TEs affect domestication? And for this talk, I'll mainly be talking about the first question, and I'll be using kind of comparative genomic methods comparing between wild species and domestic species to see any differences. And then as part of my thesis, I also want to kind of explain how the role of TEs in the evolution of domestic animals might differ to what we know in the literature to how they contribute in natural populations too. A quick aside on the data and methodology. So it's a comparative genomic framework. So I've basically collected as many domestic species as possible. Mind you, there's not a lot. There's only like 40-ish. And a lot of the wild ancestors of those as well are very difficult to find because they're just extinct. So I managed to get like 13 species pairs and then 1 triad. So that was chicken species. And that kind of represents a good diversity of different domesticates. And the step-by-step kind of methodology is collect the genomes and then verify their like domestic status and what wild ancestor they have through the literature. Then I used a pipeline called Earl Grey. I'm not sure if any tea biologists have used it, but it basically can use repeat masker and repeat modeler functionality, but it also uses custom scripts to help improve consensus sequence generation, and it's very easy to use. And then I used my own custom scripts to analyze downstream the outputs of Earl Grey. And then I used Bayesian modeling using a package called McGlim to ask those kind of fundamental questions like, does domestication have a significant effect on TE content? So this is a phylogeny of the species that I have. If you look closely, the species that are domestic and wild are paired up. So at the top you have Bombyx mandarina, and that's the wild silkworm. That clusters with Bombyx mori, which is the domestic silkworm. And then that kind of repeats that structure of the paired domestic versus wild across the whole of the phylogeny. And I've also annotated it with pathway of domestication. So the colored dots kind of show which pathway that domestic animal has undergone through the domestication process. And what this is useful for is as a proxy of kind of artificial selection strength. So with, for example, the commensal, which is blue, dogs are thought to have been domesticated under this almost accidental process where wolves kind of came in and they were increasingly tolerated by humans. So that's kind of like no human kind of conscious selection. And then directed is where when humans finally knew what domestication was and knew what phenotypes and how to do it, they then kind of applied that more consciously. So this kind of shows a gradient where directed has the most conscious human selection and probably the strongest. Commensal is the least, and prey is in the middle. And then I ran the annotations for those. So if we see, like, we can have different magnitudes of differences and in different directions for the different species, and also the kind of color coding is by the 5 major types that I discussed earlier. And there are some kind of differences in which ones are the dominant ones, and that's mostly due to phylogenetic differences. If we zoom in to the dog pair, you can see Canis lupus is the wolf and the other one is the dog. In the dog genome, there's an increase in TE content about 4%. If we consider the timescales of domestication, the dog was domesticated around 10 to 30,000 years ago. So this is quite a rapid change for such little evolutionary time. And all the others will be in the magnitude of scale of about 1,000 years, thousands of years. So this is quite quick and quite rapid. I then wanted to look at the kind of superfamily level, just to see which kind of types of TEs might actually be driving this change. So if you see on the Y, I've got the different species, again, paired up, but I've just taken a subset. And if we look again at the dog, I'm not sure if you can see the color, but the copy number kind of drives this change in L1 content, and lines are kind of indicative of the changes that you see in the overall phylogeny. I then ran Bayesian modeling, which using a package called McGlim, and basically, to kind of summarize that, it basically allows you to use like a generalized linear model framework, but account for phylogeny as a random effect, which you need because TE content is highly correlated with phylogeny. And so for this model, I wanted to ask, does domestic condition significantly affect TE content? So are domestics have more TE content or less TE content, and is that significant? And I also used genome size because genome size is known to correlate with TE content, and this was my way of introducing it as a covariate and also just to check that I did it right. So that's significant, which looks good. But the domestic condition isn't significant as this distribution kind of encounters 0, which is the dashed line. So there's no specific significant differences between domestic and wild. But if we kind of remember the phylogeny, I think that's because of the different directions of the difference in TE content for different pairs. I then wanted to partition that domestic condition factor into smaller factors. So with domestication, they're all domesticated under different timescales, different pathways like we discussed earlier. So I want to see if those might be significant and kind of look at it at a higher resolution. And again, you can see here these distributions overlap with 0, so those are, again, not significant. But that is fine, because if we look back at the phylogeny, I think this basically emphasizes the importance of different domestic conditions. So all of these will have had kind of different contexts. So they might have been domesticated at different locations, different human breeders, which select for different traits, different population sizes, and then different breeds might have been selected for after, which basically emphasizes the different conditions might have impacted them in different ways. So basically, to conclude, TE composition can change very rapidly given the timescales, and these are probably likely to factors that I was unable to kind of model because of the lack of data in the literature. And I think animals domesticated similarly have similar TE changes, which kind of provide support for that hypothesis. And a line of evidence for this is that goats and sheep have very similar parallel histories, and when you look closely, they have kind of similar directions in TE changes and similar magnitudes. And with my future work, I want to look into the associations of TEs to genes that have been under selection, for example, growth genes and immunity genes. And I also want to look at the breed formation process to see if TEs have an impact on like that level of domestication. So I'd like to say thank you to my lab mates and my supervisors, especially Toby Barrell. He developed Earl Grey, so without him, I would probably not be able to do this. But also, I just wanted to end on a quick plug. I'm finishing soon, and I did like a science illustration internship. So if anyone has any like illustration needs, please contact me. Thank you. Yep.
When you're talking about like the different pathways, given that there's so much we don't know about the domestication of these species, like how sure can you be of like your categorization of the different domestication routes?
Not sure, because a lot of what I can kind of obtain is just from the literature and what people have kind of compiled historically. So in that sense, I'm kind of obtaining this historical perspective. I think I'd like to be able to look into like different proxies to kind of establish those conditions, but it is just what it's available in the literature, and it's kind of the only available proxy I have for that selection strength question. Yep.
I'm a little bit confused about some of the species that you included, because I'm pretty sure I saw some vicunias, which
Yeah.
are known to be wild in my experience.
Yeah, so if we look back, Vicuña mensalis is like the wild ancestor, but then Vicuña pacos is like the domesticated alpaca. And so those, from my literature search, has shown to be like pacos is the domestic, and then mensalis is the ancestral version. But again, that's all on like, when I was validating them, I've compiled like a list of references that corroborate that information. And if they didn't match up, I often left them out. So these are kind of the ones I'm more sure of. Thank you.